At the beginning, we make a general assumption about our research, and we ask that the cells alter cell metabolism for their own need. And it is well known for a long time before us. But how exactly they did it, it's not our question, but how we can use it for our own purpose. And also, malignant cells have the altered cell metabolism compared to the normal one. And they also change this for their own need, for the rapid growth, for proliferation, and inviting new area of the body. As a result, we ask, and also chemotherapy work in a way that they pose a stress on the all type of cells, not only cancer cells, but also the normal cells. But normal cells more sustainable for the stress, and at the first stage, cancer cells dead or become senescent and start to proliferate. And we asked how we can combine these properties of the malignant cells, viral infection of the cells, and chemotherapy. And well known that all viruses changes metabolism of the cells in different ways, and we do not make general assumption which is the main point for all of the viruses. For example, Epstein virus increased glycolysis, lactate proliferation, flu also increased glycolysis, but flu also increased ROS signaling in the cell, disbalance in mitochondria, respiration, and any other things in the cell changing. But little is known about the enteroviruses, non-pathogenic enteroviruses. And also, it's as already mentioned, that virus make the oncolytic effect in two ways: direct lysis of the tumor cells, and also activate the anti-tumor immune response by seizing the immunosuppressing microenvironment of the cells, and also make the signals that immune cells can go like chemokines, cytokines in a new type of inflammation, also disrupting the malignant cells that present a lot of pump and many other pathogenic protein. But also, chemotherapy disrupt microenvironment of the tumor region. And this may propagate effectiveness of fire therapy in case of malignant solid tumors. And also, there's a lot of similarities between viruses infection of the cells and cancer cells. And we generally interested in promoting metabolic reprogramming of the malignant cells and tumor cells. At the first step, we decide to put our attention on the Vorobyev effect. Vorobyev effect is the effect of aerobic glycolysis in presence of enough oxygen, cells not to put oxidation of the glucose put pyruvate into mitochondria, but instead of that, they put pyruvate into lactate. This produce, there are a lot of questions why it's needed for the cancer cells, but also viruses produce Vorobyev effect in nearly in all of the cells. And what advantages give aerobic glycolysis to tumor cells? It's a rapid ATF synthesis, not so effective as phosphorylation, oxidative phosphorylation, but it also produces a lot of biosynthetic like pyruvate, nucleotide synthetase, and cell signaling changes the ROS, reprogramming chromatin of the cells, and also lactate produced go to acetyl-CoA, and this change tumor microenvironment possessing immunosuppressive region and or put the pH in a low level. But our goal of the research was to, if we put the simplest one of chemotherapy, which is well known is 2-desoxyglucose, into cancer cells, and how it will affect the replication and sensitivity to oncolytic viruses. And based on our research, we would like to present maybe in the future work in clinical therapy, and it's only beginning. In the research, we use our primary cell line. It's divided from the patient who suffered glioma 38, 21, 55, 22. It's described in picture. Stable glioma brought from ATCC, human normal cells as a control, human embryonic fibroblast, and cancer cells as a control for the activities because it's well-studied cell lines. As a virus, we use Coxsackie viruses in different stem A2, A7, A9, B3, B5, B6 poliovirus vaccine strain, and for in vivo model, we use the mice C56BL/6. Sensitivity of enterovirus. Firstly, we try to choose right virus for our research, and as it's shown from this slide, that Coxsackie B3 and B5 is more effective and do not affect normal human embryonic fibroblast. And we continue our work with for human glioma cells with Coxsackie B5, and for in vivo, because it was sensitive for mice glioma, Coxsackie B3. After that, we measure effectiveness of different concentration on drugs-desoxyglucose on the tumor cell lines, and we found that some cells respond for the therapy like we expected. This increased sensitivity, like the PDRK, 60, 67, 55, 22 cell lines, but some of the cells, U251 and 33 to 81, does behave differently. 3821 responds completely on opposite sides, the decrease in sensitivity. Then we assume that the ROS also maybe participate in the effectiveness of virus application during treatment of drugs-desoxyglucose and virus infection. And we use non-accurate antioxidant and cysteine, and we found that only for cells U251, 60, 67, 38, 21, this reduce in sensitivity to the virus infection. It make us a clue that maybe ROS participate in the effectiveness of virus infection in this cell line, but more sensitive cells like DBTG 5522 that responds to drug treatment in proper way does not affect by an cysteine treatment. We assume that maybe we can separate cells by their metabolic profiling using C-host analysis and measuring mitochondrial respiration and glycolytic capacity of the cells. For the oxygen consumption rate, we use oxygen consumption protocol from the alignment protocol. At the first time we measure basal respiration of the cell. After that we put oligomycin that can inhibit synthesis of ATP synthetase and measure the proton leak that flow through the membrane of mitochondria. Then added FCCP that encouple mitochondria and the protons start to leak freely to the mitochondria. After that we completely shut down electron transport change, and we found that surely we can divide our cells in two types, that they have low oxygen consumption rate and low mitochondrial function, and but bigger oxygen consumption rate, and this corresponds for effectiveness on 2-degree treatment with virus infection. After that we measure the glycolysis by measuring extracellular acidification rate. Like described, it's put glucose. At first, the cell treated in non-glycolytic glucose-containing media. After that, we put a glucose. Glycolysis start, but allogenecin completely shut down ATP. All glucose go to pyruvat, measuring glycolytic capacity after that, and after adding 2DG, the deoxyglucose glycolysis is shut down. And we also find that cells can be separated in two types, with high glycolysis capacity and with low one. That corresponds with the result of the effectiveness of additional DVDG and virus treatment. After that, we go to the in vivo experiment like xenograft model and using GL261 model and CT2A. After we put one injection on the viruses and put the 2-desoxyglucose in the water for the mice, and as you can see, control cells shows a rapid growth, and when we added 2-desoxyglucose virus and 2-desoxyglucose plus viruses, we can see that tumor does not grow, or we also have some mice completely recovered. In the case of CT2A, we can see that 2-desoxyglucose do not alleviate condition of the mice, maybe make it worsening. But in case of virus plus 2DG and viruses, you can see that therapy is effective. Maybe it is also corresponding for metabolic profiling of the cells. And in the last picture, I will show the mice that is completely recovered from the tumor. This only the scar that after some time completely healed. Thank you for your attention.
شكرا كثير دكتور عزام حماد.